How do cells transport molecules across their cell membranes? There are three major ways. The first we'll call simple diffusion. This is when molecules are able to diffuse right across the lipid bilayer. Only small, non-polar molecules are capable of simple diffusion across cell membranes. Examples are oxygen gas, carbon dioxide gas, and lipid-soluble molecules such as steroid hormones. Because this is diffusion, the direction of transport is always from the side of the cell with higher concentration to the side of the membrane with lower concentration. Facilitated diffusion is for polar molecules, ions, anything that can't cross a lipid bilayer membrane unassisted. This requires membrane proteins. Aquaporin is a membrane protein that allows facilitated diffusion of water molecules. So these membrane proteins are highly specific, meaning they're extremely choosy about what molecules they allow to pass. For instance, an aquaporin will allow only water molecules and not glycerol and not sodium ions or potassium ions or even protons. In addition to being highly specific, again, this is diffusion, meaning the direction of transport is from the side of the membrane where the molecule in question is in greater concentration to the side of the membrane where there is lower concentration. So for facilitated diffusion, it requires membrane proteins. These membrane proteins may form channels or actually act as carriers for these molecules. There is no energy involved here. They're highly specific, and molecules are transported down their concentration gradient. What if a cell needs to actually transport molecules against a concentration gradient or uphill? Well, that requires energy. So active transport always requires energy, and again involves membrane proteins. And it's the membrane proteins that are actually doing the coupling. They couple an energy releasing process like ATP hydrolysis to the energy expensive process or energy consuming process of transporting molecules against their concentration gradient. Again, active transport mechanisms are highly specific. So how do we tell the difference between facilitated diffusion, for instance, and simple diffusion? Neither of them require energy, and both of them have transport in the same direction. The key is that facilitated diffusion requires membrane proteins, and there's a limited number of membrane proteins for the facilitated diffusion of any particular molecule. And we can illustrate the difference by plotting the kinetics of transport. Here, this is going to be a plot of the transport of molecules into the cell against the concentration difference across the membrane. When there is no difference in concentration across the membrane, there is no net transport. Now, first, let's look at simple diffusion of a molecule like oxygen. As the concentration of oxygen outside the cell increases relative to the concentration of oxygen inside the cell, you will get faster and faster oxygen transport. And this is going to increase pretty much linearly because oxygen molecules are crossing the lipid bilayer membranes. The more there is, the faster they can get in. Now, what about something that requires facilitated diffusion? Well, a molecule that comes in by facilitated diffusion is, again, it's going to show initial linear kinetics. The higher the concentration difference across the membrane, the faster the rate of transport through facilitated diffusion. But what you'll see is that as the concentration gradient becomes higher and higher, or the concentration difference gets greater, the rate of transport begins to plateau. So this is facilitated diffusion. And it begins to plateau at a region where now every single channel or carrier molecule is fully occupied. I like to use the analogy of people trying to get into a football stadium. The protein channels are like the turnstiles or the gates. When there's only a few people trying to get in, the number of gates is not limiting. As more and more people try to get in, you get faster and faster movement into the stadium. But at some point, when you have hordes of people trying to get in, every turnstile or gate is busy, and the only way that you can get faster movement of people into the stadium would be to open new gates. So in this plateau region, the rate of transport into the cell is now limited by the number of available channels or carrier molecules. So all of these protein-limited processes show what we call saturation kinetics. Now, how would the kinetics of active transport, if you plotted the kinetics of active transport, how would that look? And I'm going to leave that to you as an exercise.